Good day. I am Gerion Izaak Manalo, and today I will be discussing viral evolution. For our introduction, the high mutation rates of viruses, coupled with short generation times and large population sizes, allow viruses to rapidly evolve and adapt to the host environment. Viruses evolve within a host during transmission to novel susceptible hosts and establish infections in new host species. High mutation rates create many viral variants. Furthermore, the unique ability of viruses to change allows them to cross species barriers, resulting in zoonotic infections. Virus evolution is further characterized by additional layers of complexity. One unique characteristic of viruses is their MOI, or multiplicity of infection, which is the ratio between the number of viruses and the infecting cells. The typical view of viral evolution is that viruses create huge population sizes within the infected host. Next, let us proceed to mutation and substitution rates in viruses. As we can see, this is the table of examples of range of mutation rates and evolutionary rates of some viruses. So, what is mutation rate? Mutation rate is typically defined as the average number of errors created in genomes of viral progeny per base per replication cycle. This rate changes from approximately 1.5 by 10 to the negative third power in the RNA bacteriophage up to 10 to the eighth power in the DNA virus herpes simplex. So how do we measure the mutation rates? Mutation rates refer to the rate of mutation per site per genome replication, right? Or the rate of mutation per site per round of viral replication. Critically, there are two types of measures that we use, and these are the stamping machine replication and geometric genome replication. The stamping machine replication uses one single virus genome as a template for replication, leading to linear accumulation of mutations. Whereas in geometric genome replication, progeny strands can become templates for replication themselves, and thus there is an exponential or geometric increase in progeny genomes. And this will lead to a completely different distribution of mutations in the progeny genomes. This is an illustration of the two replication measures. We must remember that high MOI, or multiplicity of infection, leads to a myriad of complex and contrasting effects. And what are these? To summarize, it is evident that high MOI leads to complex and conflicting effects on genome selection. For example, high rates of recombination or reassortment may also lead to the emergence of strains with a more virulent phenotype, and as is thought to occur in cross-species transmission events of influenza virus. High MOI also produces a higher gene copy number, that is multiple genomic copies of the same gene in one infected cell. Interestingly, the population with highest fitness in the original host does not necessarily fare well in new hosts, while low-frequency genotypes from original host may be the ones that prevail in the new host. This means that if the population is fit, the virus does not stay on the population. But with the population that has a low frequency of genotypes, then they may become host and the virus may prevail in them. And what are the other implications of MOI? There are three. Number one, the distribution of viral particles at different sites of an infection is unknown and will also directly affect MOI and the efficiency of selection. Number two, the MOI and ensuing diversity of the transmitting population affect the probability of establishing an infection in a novel host. And the last one, it is likely that different types of viruses will be affected differently by MOI. So it is really said that the effects will be contrasting and sometimes different from each other. Phylogenetic. So what is phylogenetic? Phylogenetic methods are rooted in the powerful methodologies of phylogenetics, which emphasize the phylogenetic tree as a key to investigating evolutionary process. The phylodynamic approach has yielded remarkable insights into viral evolution. As an example, Hu, 1995, collected a set of isolates of poliovirus from a 10-year sequential infection chain in South America and used this to determine the number of mutations per year, which is uh, e- equivalent to 9 times 10 to the negative 3 nucleotide substitution per site per year for a 150 nucleotide window within the 7,500 nucleotide genome. When the nucleotide difference from the ancestral sequence of each extant sequence is plotted against time, it is possible to infer the rate of nucleotide substitution and thus track back the date at which the ancestral sequence emerged. And we also use molecular clocks to measure and determine where the virus originated. We can also see in this graph that most of these changes were synonymous mutations that would have little, if any, influence on the biological properties of the virus. Holmes suggested that a number of different patterns can explain viral dynamics across the globe, and these are the examples. Number one, random mixing, or the frequent viral traffic among different regions of the world, cause lack of spatial structure in the tree, with no correlation between the geographic location of a sample and its location in the phylogeny. The second one is the population division, in which strong spatial subpopulations of viruses localize to certain regions of the world, suggesting that these populations do not mingle. And the third one is source sink transmission, where one viral population acts as the source for all other viral populations in the world. A vast experience with antiretroviral drugs for the treatment of HIV has shown that the virus swarm contains about 10 to the negative 5 variants, and that is a huge range of variants of virus for HIV that will resist any single drug. With this said, many such mutants will be produced each day in an HIV-infected patient. For this reason, it has been found that effective control of HIV infections in humans usually requires the simultaneous use of at least three drugs, each of which requires a different set of mutations to escape drug control. For our conclusion, it appears that viruses have used many strategies to perpetuate themselves in their host populations. Some strategies are benign or it has no effect, while some others cause serious disease in their host. We have learned that viruses really do their best or make effort to mutate and perpetuate themselves or change according to the environment of the host so that they may stay and penetrate the host. Thank you for listening. This is a short topic, and thank you.